Hello everyone, how are you all? I hope you are doing great. So today I will discuss about the very interesting topic, that is variant classification according to ACMG-AMP guidelines. So before starting, I would like to tell you about CleanVar database. I think many of us know about it. It is the largest freely accessible public database for variant interpretation. It is in collaboration with DBSNP, PubMed Central or RefSec databases. This database provides the information about the variants, their associated disease and interpretation history. In this database, any researcher, clinician or genetic counselor can submit their variants which they have found in their research, and anyone can access those variants and their interpretation criteria. Let me show you the variants which I have submitted in CleanVar. Okay, in total, I have submitted 41 variants which I have found in my research along with their health status and interpretation. So these are the genes in which I have got the variants. These are their associated disease, and I have followed the ACMG 2015 guidelines for interpretation. So in this particular video, I would like to share my experience that how I interpreted or classified my variants. In a few decades, sequencing techniques have been gradually developing. Like in 1970s, first generation sequencing came, that is the Sanger sequencing. That was manual at that time. After that, in 1990s, automated Sanger sequencing options were opened. After that, in 2000s, second and third generation sequencing, that is the next generation sequencing came. So as we know, through all of these sequencing methods, we get the variants in our particular patient. But what after the sequencing? What is the utilization of these variants if we do not know if they are disease causing or not? So next question comes, how to interpret the variants if they are causing disease or not? That is when ACMG-AMP guidelines for variant interpretation came into picture. So ACMG stands for American College of Medical Genetics and Genomics, and AMP is Association for Molecular Pathology. Actually, before ACMG-AMP guidelines were provided, there was a lot of confusion about the variant annotation. Means a single variant had multiple annotations. Why? Because different geneticists or clinicians were using different methods to interpret the same variant. Some were using case control studies, others were using annotated database or segregation studies, population database, computational methods, etc. So these different methods leads to different annotation. So in 2013, ACMG assembled a work group which included the representatives from ACMG, AMP, and College of American Pathologists to revisit and revise the standards and guidelines for variant interpretation. These guidelines were approved in 2015. So main goal of ACMG-AMP guidelines was to provide a consistent standard way to create a framework to determine the pathogenic variant within a gene that has a role in Mendelian disorder. So this is the paper. This is the research paper by Richards et al., which described ACMG criteria in detail. So what was the result of that meeting? Guidelines suggested that the term mutation and polymorphism led confusion in interpretation whether they are disease-causing or benign. Actually, mutation was defined as the permanent change, while polymorphism is a variant which is found in greater than 1% of population. So both the terms were replaced by variant. ACMG classified variant in 5 categories, that is pathogenic, likely pathogenic, uncertain significance, likely benign or benign on the basis of 8 data sets, which I will discuss later. So ACMG suggests that the term likely pathogenic or likely benign are used when there is greater than 90% certainty of a variant either being disease causing or benign. Means if we are saying that a particular variant is pathogenic, then there is greater than 90% chances of that variant to be pathogenic. So ACMG combines the 8 data sets and suggests the 28 standard stringent guidelines to correctly interpret the variant, and they have formatted these guidelines in 28 attributes. And the main thing which I would like to highlight is that these attributes were designed for variant interpretation in Mendelian disorders, means single gene disorder. Okay. So these guidelines define 28 standard criteria that address various type and level of evidence to interpret the variant. These standard guidelines represented by 28 attributes which were classifying the genetic variant in Mendelian disorders. These 28 attributes were divided into two categories, pathogenic and benign. Pathogenic attributes were further divided into 4 categories based on the evidence. Means if evidence for pathogenicity is very strong, it will be denoted as VS. For strong, S. For moderate, M. And for supporting, P. Likewise, in benign, if it is standalone, it will be denoted as A. If evidence is strong, it is S. And for supporting, it will be P. So now we will see how this attribute has been named. Like PVS1, P for pathogenic, VS for very strong, and 1 denotes the number of guidelines followed to interpret that variant. Similarly, for PS1, PS2, PS3, PS4, means the integer denotes the number of guidelines followed, like PM1, PM2, PM3, PM4, PM5, PM6, and so on. Likewise, for others, for benign case, it is BA1, benign, A for standalone, and 1 is the guideline followed to interpret that variant. So in this way, these are the 28 attributes in total. So now, how to apply these 28 attributes for interpretation, which seems quite confusing? Let's resolve it. These are the 8 data sets which ACMG used combinedly to interpret the variant, and on the basis of these data sets combinedly, they classified a variant into 5 categories. So I will discuss these 28 attributes in detail in next few slides. As we saw, pathogenic variants were classified on the basis of very strong evidence of pathogenicity, strong evidence, moderate, and supporting evidence. So first comes PVS1. It includes null variants like nonsense, frameshift, deletion, or splice site variants, which are up to 1 or 2 upstream or downstream the exon, and where the loss of function mechanism of disease has been established. So we will check the gene in which we are getting variants that it has loss of function mechanism or not. I will discuss LOF, loss of function mechanism, and null variants in detail later. Okay. So caution we should keep in mind with this particular class are that we should exclude the genes where loss of function has not been established, like splice site variants which are predicted to lead to exon skipping but leave the remainder of protein intact. So next classification was based on the basis of strong evidence of pathogenicity, and first class is PS1. PS1 includes the synonymous variants which are published as pathogenic variant in literature. Means nucleotide change can be different or same as the previous established variant. PS2, it includes de novo variants where paternity and maternity is confirmed in that particular patient. De novo variants are those which are found in an individual for the first time. Means they are not inherited from the parents. Then PS3, it includes the variants in which in vitro and in vivo functional studies have confirmed the damaging effect of variants. PS4 includes the variants where prevalence of that variant is slightly increased in patients as compared to controls. The next criteria is based on moderate evidence of pathogenicity, and it includes PM1 to PM6. So PM1 includes variants which are present in mutational hotspot. Hotspot means that gene segment is prone to genetic alteration, or at well-established functional domain where benign variation is not found. Then PM2, it includes variant which is not present in control database. PM3, where variant is detected in trans with pathogenic variant and the disorder is recessive. PM4, it includes stop loss variant or variant which cause protein length changes due to in-frame deletions or insertions in a non-repeat region. Then PM5, it includes novel missense change at amino acid residue where a different missense change was found to be pathogenic. For example, arginine 156 histidine, it is reported as pathogenic, and now we will observe arginine 156 cysteine. So here arginine is getting converted into histidine, and here arginine is getting converted into cysteine. So if this variant is pathogenic, then this variant will also be pathogenic and it will come under PM5 criteria. Then comes PM6. It includes de novo variants but without confirmation of paternity and maternity. The next criteria is based on supporting evidence of pathogenicity, which includes PP1 to PP5. So PP1, it includes variants which are in co-segregation with disease in multiple affected family members in a gene which is known to cause the disease. PP2, it includes missense variant in a gene that has less number of benign missense variants and where missense variants are the common mechanism of disease. Means that particular disease is causing by that particular missense variant. Then PP3, it includes variants where various computational evidences support a deleterious effect on gene product. PP4, when patient's phenotype or family history is highly specific for a disease with a single genetic cause. And a variant will be put in PP5 category if reputable source recently reports variant as pathogenic, but the evidence is not available to perform the independent evaluation. Now coming to criteria for classifying benign variants. On the basis of standalone criteria, if the allele frequency is above 5% in population database for any variant, it will be put in BA1 category. Then based on the criteria of strong evidence of having benign impact, it will be classified into 4 categories. First is BS1, which includes the variants with frequency more than expected. BS2, it includes variants observed in a healthy adult individual for a recessive, dominant or X-linked disorder with full penetrance expected at an early age. Full penetrance means if all individuals who have the disease causing variants have clinical symptoms of disease. Then BS3, when well-established in vitro or in vivo functional studies show no damaging effect on protein function or splicing. BS4, it includes variants in those there is lack of segregation in affected members of a family. On the basis of supporting evidence of benign impact, this category includes 7 classes. BP1, which includes missense variant in a gene in which mostly truncated variants are known to cause the disease. BP2 includes variants which are observed in trans with a pathogenic variant for a fully penetrant dominant disorder, or observed in cysts with a pathogenic variant in any inheritance pattern. In-frame deletions or insertions in a repetitive region without a known function are included in BP3 category. BP4 includes the variants where various computational evidence suggests no impact on gene product. When a variant is found in a case with an alternate molecular basis for disease are included in BP5 category. BP6 includes reputable source reports variant as benign impact, but evidence is not available to laboratory. Then that variant will be put in BP6. BP7 includes synonymous variant for which splicing prediction tools predict no impact to the splice consensus sequence nor the creation of a new splice site, and nucleotide is highly conserved. So these are the 3 points to remember while interpreting a variant. So we should know, is the variant or gene is associated with patient's clinical profile? Or we should know the mode of inheritance of that variant. That how is this variant inherited? Is it autosomal dominant, autosomal recessive, X-linked or de novo? And last, zygosity of variant. We should know about the zygosity. Is it homozygous, heterozygous, wild type or compound heterozygous? Then we will be able to interpret that variant. So now we will discuss some points or questions that will help in correct interpretation of a variant, and it will make the interpretation easier. So first is the variant, we should know before the interpretation that the variant is located in a conserved region of the gene. If yes, then that will be pathogenic because variants in these regions are more likely to be pathogenic. Second, is the variant predicted to disrupt protein function? Okay, so variants which predicted to disrupt protein function are more likely to be pathogenic. Third, has the variant been previously reported in people with the disease? If yes, then it will be pathogenic. If a variant has been previously reported in people with the disease, this is a strong evidence of pathogenicity. Then fourth comes, is the variant found in multiple affected individuals from the same family? If a variant is found in multiple affected individuals from the same family, this is the strong evidence that this variant will be pathogenic. Okay, so we should know, is the amino acid change located in a critical domain of the protein? If yes, then there are more chances of that variant to be pathogenic because critical domains are essential for protein function. We should know, is the amino acid change is conservative or non-conservative? If it is conservative, there are more chances of this variant to be benign because conservative changes are less likely to disrupt the protein function, while non-conservative changes are more likely to be pathogenic. Then, is the amino acid change found in a known disease-causing hotspot? If yes, then there are more chances of this variant to be pathogenic because hotspots are regions of the genes that are more likely to contain disease-causing variants. So by asking these questions, we can gather the evidence needed to classify a genetic variant as pathogenic, likely pathogenic, variant of uncertain significance, likely benign, or benign category. So these are the 8 data sets and these are their associated attributes. In this particular video, we will study about the variant type and their attributes. Some of the variants affect protein structure or function and some do not have any effect on the protein. So on the basis of effect on protein structure or function, variants are of various types. Like first is synonymous and intronic or intergenic variants, which have low impact on protein structure and hence its function. So synonymous variants are variants in which there is no change of amino acid. If we see here, this is the normal sequence and this is the altered sequence in which C is replaced by A. So ACC and ACA both codes for threonine. So there is no change in amino acid and hence no change in protein folding. Second is intronic or intergenic variants. Mostly they don't affect the protein as they do not encode the protein. So they can be considered as having benign effect. Second category includes the variants which have moderate effect on protein. And it includes non-synonymous variant in which there is change of amino acid. If we see here, A is replaced by T. While ACC codes for threonine, but TCC codes for serine. So the effect will depend on the site of variant. If this variant is present at functionally important site or not. If it is present, it may cause pathogenic effect, and if it is not present at important site, it will not cause that much effect. Next is non-frameshift variant. When there is addition of only one amino acid, or we can say 3 nucleotides. Then the frame of 3 nucleotide is intact after that added amino acid. So effect again will depend on the if that change is present at functionally important site or not. Then third category includes the variants which have high impact on protein, and it includes frameshift and stop gain variants. In frameshift, there is addition of a new nucleotide which will shift the frame of 3 nucleotides or one amino acid. As we see here, there is addition of one nucleotide, that is T, that will change the whole protein after that. And next is stop gain variant. If we see here, there is change at this nucleotide where A is replaced by T nucleotide, and that is the stop codon TGA. So due to change of this nucleotide, protein formation will stop here and protein will be truncated. That surely have high impact on protein and that may cause the disease. So these are the variant types. On the basis of them, we can interpret our variant of interest as different type of variants have different impact on protein. So now we will discuss all these attributes which are associated with variant type. So first is PVS1. As we discussed earlier, P denotes pathogenic, VS very strong, and 1 is guideline followed. So null variant is when there is complete absence of gene product which forms the truncated protein. Null variants include any variant that leads to NMD, means nonsense mediated decay of the transcript, by preventing ribosome from reaching the last coding exon junction. So what is nonsense mediated decay of mRNA? So we will see here, this is the mRNA, this is the start codon AUG, and that is the stop codon. So after splicing, all introns are removed and we get this exon junction complex at the site from where the introns were removed. So in normal condition, when translation occurs, this exon junction complex is displaced by ribosome and protein will form till ribosome reaches the stop codon. This is the normal scenario. But when there is stop codon before its actual site, this is the transcript with premature stop codon. It should present here, but the stop codon is present before its actual site. So what will happen in this condition? ribosome will stop at this position instead of this. So EJC means exon junction complex will not displaced and protein formation also will end here before the actual stopping position. So protein will be truncated, and that is called the nonsense mediated decay of mRNA. So about null variants, we have to remember certain things like all the null variants have same functional consequences, like they all leads to absence of gene product. Okay. And the relevance of a null variant depends on the disease mechanism of the gene. Means a novel null variant in loss of function gene will be classified as pathogenic, but this method requires an abundance of clinical evidence to support that the loss of function mechanism is present or not in that particular gene. For LOF genes, if premature termination codon is upstream of a variant that has been previously classified as pathogenic, then the novel variant clearly also truncates then that the essential part of the protein. Now the question comes, if we have different type of variants, then for null variant, it is clear that it will put in PVS1 criteria along with the conditions applied, which I have discussed earlier. But what if they are missense, splice site, silent or intronic variation? So let's move forward. Splice variants can also lead to the loss of the gene product. But in that case, there should be strong evidence that this transcript is 100% affected by that variant or not. And variants in the highly conserved donor GT or acceptor AG nucleotides are very predictive and considered strong evidence toward pathogenicity if loss of function mechanism has been established. And splice variants in the intron upstream of the last coding exon are treated using the criteria for affected exon preserves reading frame and are treated as truncations. Then missense variant and in-frame indels. Missense changes may or may not affect the protein, and the consequence of a missense variant depends on the nature of the amino acid change or indirect information about the presumed importance of the original residue. So relevant indirect evidence includes information about the consequence or association with the disease of other missense changes affecting the same residue, or the knowledge about the function of the residue in the context of the 3D protein structure. Similarly, small in-frame indels are also treated with the same fundamental uncertainty. So if a novel missense change or deletion affects residues shown to harbor other pathogenic missense changes or that are determined to be structurally essential, it is more likely to be pathogenic. Then start loss, stop loss variants, silent or intronic variants. Start loss variants are missense changes that affect the initiator methionine, which are likely pathogenic. Because the possibility of alternate isoforms or alternate translation start sites cannot be easily ruled out, the value of this variant type is limited and such variants will remain classified as variants of uncertain significance in the absence of evidence of any functional or additional clinical data. Similarly, stop-loss variants are treated extremely carefully as many proteins can tolerate the additional sequence. As in stop-loss variants, there is addition of sequence. Silent and intronic changes do not exert an obvious effect on transcript stability or protein sequence. So it may be possible that such variants are pathogenic, but they are less likely to exert an effect on gene products and therefore less possibility to lead to the disease. Now, first question comes, if we found a synonymous variant, then various possibilities can occur. First, if that synonymous variant is same as the previously established pathogenic variant, regardless of the base change, means it doesn't matter if nucleotide changes or not, that particular synonymous variant will be considered as pathogenic and put in PS1 category. Then second condition, if algorithms predicted no impact on splicing by that particular variant and the nucleotide change is not highly conserved, then we can put it in BP7 category, means benign category. Now, if we found in-frame insertion or deletion variant, then we have to check if that variant is found in non-repetitive region and that change the protein length, then we can put it into PM4 category. And if it is found in repetitive region without the known function, then it can put in PP3 category, that is benign. Now, if we found missense variant and the missense variants are the common mechanism for causing that particular disease, then we can put it in PP2 category, that is pathogenic. And if we have found missense variant, but for that truncating variants are known to cause the disease, but not the missense variant, then that variant will be benign under category PP1. And if we found the novel missense variant at the position where previously a different missense change has been reported pathogenic, so that will be put in PM5 category. So this was all about the variant type and their attributes. Okay, we have covered all the attributes related to variant type, and in the next video, we will learn about the population and computational data sets and their attributes. And then we would be able to answer these following questions. So see you in next video. Thank you very much.